It's over, ma'am, for them. DL people, actually DL people, I have completed the syllabus for one batch. Some 6 students are studying.
How many months will it take for a complete course?
Minimum 3.5 to 4 if we take continuously. Months, of course. It's vast, no? Okay. Okay. Good evening, everyone. Please confirm me. Am I audible to everyone? You two people, please confirm me. Am I audible to everyone? Just type in the chat. I can sit here. Can anyone confirm me? Am I audible to all of you? Nani, please confirm. Am I audible to everyone? Wonderful. Let's continue. Okay. Everyone, please confirm. All 20 people. Am I audible? Okay, perfect. Let's continue. Okay. As we discussed in the last class, so today I will be starting the cell biology topic first. Okay, I think we have done the complete syllabus analysis. For the zoology also I will do the syllabus analysis on the Monday, and we will do the continuously. Whenever we consider the syllabus which is common for the both botany and zoology, is there are few chapters, that is cell biology, molecular biology, genetics, biochem, rDNA technology, ecology. Okay, so these are the topics which are completely common, compared to the botany and zoology. Compared to the botany, in case of zoology we will get more applied questions. When it comes to the botany, we will get the questions most of the time statement based questions. So all the students has to read the complete cell biology from very basic to the very depth. How many number of questions that we can expect exclusively from the topic of the cell biology? If we consider the last Gurukul DL paper and the papers of the 2016, let's consider the recent papers. You get an average of 12 questions and maximum of 15 questions can appear from cell biology topic. Now everyone please listen for 30 minutes. After that you people can raise your all doubts. What exactly we have to focus in the cell biology chapter? So listen, we have to focus on all the organelles starting from plasma membrane. I am talking about the most important organelles where the questions will be repeatedly asked. Apart from the plasma membrane, we have to focus on nucleus, mitochondria, lysosomes, peroxisome, and the most important organelles, that is RER, SER, then Golgi complex and cytoskeleton. Trust me, the most difficult question only comes from these 4 specific organelles. It doesn't mean that we have to leave all other organelles, we have to read all other also. And from which topic or which organelle we get the question compulsory apart from these? Plasma membrane. In the plasma membrane we have to read the structure of the plasma membrane, composition of the plasma membrane, type of proteins, type of lipids, type of carbohydrates and their functions we have to read very, very elaboratively. Now when we consider apart from the cell organelles, what we have to focus on? Apart from the cell organelles, we have to focus on cell cycle and its regulation. Minimum 2 questions you can expect from here. Most of the students will just read the mitosis and meiosis and they won't focus on the regulatory part. The difficult question always appears from the regulation. Apart from this, another very common topic which is they use it to repeat frequently, that is chromosomal organization, where we have to read about the heterochromatin, euchromatin and a very specific topic named as the transposable elements or the transposons. These are the various topics that we have to focus on or focus in the chapter of cell biology. I am not giving you the 100% assurance, but 90% of the questions undoubtedly will comes from the area that what we are going to discuss. What you have to do after completing the lesson, first watch the video, keep the PPT, and along with the PPT, whatever the reference books that I will provide for the specific chapter, just read them thoroughly. Then automatically you can answer any kind of a question that is coming from this topic. All right. Now everyone listen to me 30 minutes. After that I'll give the time to ask your doubts. Let's start plasma membrane. Whenever we are starting the plasma membrane, we have to focus on structure. Most important friends, we have to focus on the composition of the plasma membrane. The last part is functions. When we consider the functions, plasma membrane in general involved in the transport. We will see all those. Now, let's understand the structure of the plasma membrane. What are the major reference books that we have to cover for this particular cell biology? We have to cover Karp, Bruce Alberts, Johnson and Alberts, and the groundbreaking book, that is the Lodish. You do not have the time to cover all these topics, hence you can follow the PPT thoroughly and you can follow the reference material that what I will give to all of you. Now let's understand plasma membrane. Tell me everyone, how can we give the simple definition of the plasma membrane? We generally use it to say that it is selectively permeable. Why we use it to say it is selectively permeable? Because it never allow every molecule, every ion to cross the membrane. Whatever the ions that it has to transport, it selectively choose it and it will transport only those molecules. Can you see here, plasma membrane generally we call it as container for the cell. Why do we use the word call it as container for the cell? See, can you see here, we have written it holds the cytoplasm and all the organelles together. Let's consider friends, this is the cytoplasm. What is cytoplasm? Just a gel-like structure, and on the cytoplasm all the organelles are present. Generally this cytoplasm is covered by plasma membrane. If it is a plant cell, the plasma membrane is covered by the cell wall. So what you have to understand, so here plasma membrane acting like a container which is holding all the cells and the cytoplasm. That's why we call it as the container for the cell. And why we call it as the barrier for the cell, since it is involved in the permeability of the selective ions and selective molecules. That's the reason we call it as the barrier for the cell. In order to explain the structure of the plasma membrane, there were many theories were proposed. Among those, a simple theory which is proposed by a Garter and Grendel in the year 1925. You know what they said? They said that plasma membrane might contain a lipid bilayer. They have not concluded the statement also, they just said that it might contain the lipid bilayer. So, but you people know that plasma membrane generally contains what? Proteins. Plasma membrane generally contain cholesterol. Plasma membrane generally contain carbohydrates as well. But what they said, it might contain lipid bilayer, but they have not concluded that apart from the lipid, is there any other molecules are present on the membrane or not. Anyway, even though they proposed that they might contain a lipid bilayer, they have not proved. There is no experimental evidence to prove that it contain, the plasma membrane only contain lipids. What you people can observe, lipid head and tail. Lipid head and tail. They are arranged in a two layers. That's the reason they call it as the lipid bilayer. But of course, this theory was not accepted because there is no proper evidence to prove that plasma membrane only contain lipid bilayer. Then after Garter and Grendel, everyone remember, in the year 1935, Davison and Daniel, they proposed a model called as a sandwich model. You know what is the sandwich model? If everyone quickly recall, if I want to prepare a veg sandwich, how does it look like? We keep two breads. In between the breads, we will keep our veggies or meat. That is how the sandwich look like. Same thing. If you observe the lipid bilayer is present in the middle. and this lipid bilayer on either side, it is covered by a layer of proteins. So that's the reason we call it as what model? Sandwich model, in which the phospholipid bilayer lies between two layers of globular proteins. That's what we can call the sandwich model. But what you people can observe, according to this model, only proteins are located on the top and lower layer and plasma membrane is located in the middle. So this is also not correct model, which did not explain the proper structure of the plasma membrane. So this is proposed in the year of 1935. Then everyone listen, later in the 1972, you all people read multiple times, Singer and Nicholson. They proposed that this is the correct model. Listen everyone, they proposed that membrane is a mosaic of proteins dispersed within the bilayer. What is the meaning of mosaic? Mosaic means group, variety of proteins dispersed within the bilayer. They also said that, icebergs on the sea. This is what they proposed. Suppose example, let's consider, if you observe the ocean, the starting of the ocean is what? It is full of the water, correct? If you go deeper of the ocean or the middle of the ocean, what you people can observe? You can observe the icebergs on the top of the water or in the middle of the water. Same way it look like. Now you just consider entire lipid bilayer looks like this. On the top of the lipid bilayer, lot of proteins are embedded within the membrane, and all these proteins are not belonging to the same group. All the proteins are of different types. That's why we can call it a mosaic, or a variety or a different group of proteins embedded within the plasma membrane. Now every student pay attention from this particular slide. According to the Singer and Nicholson, this is how the plasma membrane look like. Every one of you focus on the membrane. How does the plasma membrane look like? It contains a lipid bilayer. On the lipid bilayer, what are embedded? Can you see that? Lot of proteins are embedded. Can you see here? This is a type of a protein attached to the hydrophilic head of the lipid. This is called hydrophilic head. To the head of the lipid, these proteins are attached. These kind of proteins, we generally call it as what kind of proteins? Peripheral proteins. Why do we use the word peripheral proteins? Because it is embedded on the periphery or end region of the plasma membrane. That's why we called peripheral proteins. Now, if you look into the second type, what you people can observe with this kind of a protein? Integral membrane protein. What is the meaning of an integral membrane protein? Which is embedded within the plasma membrane, which is embedded in the deep regions of the plasma membrane. Can any one of you give me the example of the integral membrane proteins? See, on the plasma membrane we have channels through which ions can move. We have channels, carrier, pumps which are present on the plasma membrane. They are strictly integral membrane proteins which are embedded within the plasma membrane. Now, we have a peripheral proteins, we have a integral proteins. We also have transmembrane proteins. What are called transmembrane proteins? Which are transpanning, which are moving on the plasma membrane. That is what exactly we can call the transmembrane proteins. Now, on the plasma membrane, peripheral, integral, transmembrane proteins are present. Now, what else the plasma membrane contains apart from the lipids and proteins? Apart from the lipids and protein, plasma membrane contain abundant amount of carbohydrates, which are sugars. You read in the 10th and 11th standard, whatever the sugars that are present on the plasma membrane, they never present directly on the membrane. Either they will be attached to the proteins present on the membrane, or they will be attached to the lipids present on the membrane. You all read, right? Whatever the sugars that are attached to the lipids, we call it as what kind of a lipids? Glycolipids. Why do we call glycolipids? Where the sugars are attached to the lipid components, we call it as glycolipids. and sometimes sugars are directly attached to the protein. Correct na? Why it is directly attached to the protein? Because they will attach to a specific linkage. Those are said to be what? Glycoproteins. There was a question asked in the exam, which kind of a sugars are most predominant on the plasma membrane? Obviously glycoproteins, which means, an example I am saying you, let's consider 100 sugars are there on the plasma membrane. In that 100 sugars, mostly 80 sugars are strictly attached to the proteins only. That's why glycoproteins are more abundant compared to the glycolipid. So plasma membrane contain lipid bilayer, plasma membrane contain proteins. Apart from the lipids and protein, what else the plasma membrane contain? Plasma membrane contain cholesterol. What is the function of cholesterol on the membrane? What's the function, everyone? What is the major function of the cholesterol on the plasma membrane? Everyone, online people. Very good. So whatever the cholesterol that is present on the membrane, it is responsible for fluidity of the membrane. Never ever think and never forget that plasma membrane will never be stagnant. Plasma membrane always moving. So that is what we exactly call it as fluidity. That fluidity of the plasma membrane given by what? Cholesterol molecule, which are a type of sterols. This is the simplified structure of the plasma membrane. Now what we have to read for the exam? We have to read what are the types of lipids are present on the membrane, what are the types of proteins are present on the membrane, and what are their functions. Then very important, type of carbohydrates present on the membrane and their function. Now there is a simple question. Every student please answer, all the online people. Among the lipid, among the protein, among the carbohydrates, which one will perform maximum functions of the plasma membrane? Which one will perform maximum function? Which will be present in the abundant quantity? Quantity. Quantity wise will be the lipids, function wise will be the proteins. Now again there is a simple question, among the lipids, protein and carbohydrates, which plays a key role in the cell-cell communication?
Carbohydrates.
Everyone, very good. Sugars majorly plays a key role in the cell-cell attachment, cell-cell communication also. Very good. Now everyone just focus. What we understand? What is fluid mosaic model? Where mosaic means a group or variety of proteins which are embedded on the fluid. What is fluid here? Lipid. Lipid always appear as a fluidized structure. That's why we can call fluid mosaic model. So undoubtedly plasma membrane contain variety of lipids. Among all the lipids, which lipids are more predominant? Undoubtedly phospholipids are the most abundant lipid. Now this statement is not a universal statement. Why? Because different cell membranes contain variety of lipids. Suppose let's consider neuron. Neuron is not rich of the phospholipid, neuron is rich of the glycolipids. So everything is dependent on the type of the cell, right? Now, but in general phospholipids are more predominant. Now phospholipids forms a bilayer. Whenever you observe the structure of the phospholipid, how does it look like? Phospholipid contain, everyone look into the picture. Sorry. Phospholipid contain what? A hydrophilic head and a hydrophobic tail. You already read multiple times in the 11th standard only. When you observe the hydrophobic tail, it is made up of what? Both the saturated fatty acids are also and unsaturated fatty acids also. Why unsaturated fatty acids are bent? What is the reason? Because of the double bond, they may bend or they can form a kink-like structure. That bending is generally called as kink. Now the most important point you people has to remember, wherever the bent or kink are present, their fluidity will be more. So undoubtedly, compared to the saturated fatty acid, unsaturated fatty acid will provide more fluidity. Now what kind of lipids we have to focus? Now first we will focus on lipids and we read extreme depth. We have to focus extreme depth of proteins and carbohydrates, where we people can expect the questions in the exam. Let's consider the lipids first. We have to focus on phosphoglycerides, we have to focus on sphingolipids, we have to focus on cholesterol, right? Now let's consider the number one, phosphoglycerides. How can you give the definition of a phosphoglycerides? Everyone look into this. Most membrane lipids contain a phosphate group, in which because of the phosphate group we call it as phospholipids. Then why we called glycerides? These phospholipids present or lies on the glycerol as their backbone. That's the reason we call it as what? Phosphoglycerides. Every one of you look into this picture. If you observe the picture carefully, this is the hydrophilic head. What the hydrophilic head contain? Phosphate group. And that phosphate group is embedded on the glycerol as their backbone. Correct na? I strongly believe everyone know about it, triacyl glycerol. What is triacyl glycerol? The stored form of the lipid. Triacyl glycerol made up of how many glycerols? 3 glycerol, which means 3 OH groups are involved. But when you consider plasma membrane specifically, plasma membrane basically contain diacyl glycerols or monoacyl glycerols. Most of the triacyl glycerols are stored forms, whereas diacyl glycerol and monoacyl glycerols are structural forms. No need to get confused over there. Now, since phosphate group is embedded on the backbone of the glycerol, we call it as the phosphoglyceride. Now, don't get confused here. I'll explain it very slowly. Everyone just try to understand. Okay. Now, phosphoglyceride, if it, suppose let's consider phosphoglyceride. Okay. Now listen. Anyway, I will explain in the functions also, but remember, if the phosphoglyceride, if it is having a head group, what group? Head group as the choline, we call it as the phosphatidyl choline. If the phosphoglyceride, instead of the choline, if it is having a serine as their head group, we call it as phosphatidyl serine. Instead of the serine, if it contain inositol as their head group, we call it as phosphatidyl inositol. If it is having ethanolamine as their head group, we call it as PE. So it's mandatorily, you don't need to remember the structure. I want you to compulsory remember the type of the phosphoglycerides. Phosphatidyl choline, phosphatidyl serine, phosphatidyl ethanolamine, phosphatidyl inositol. How did we categorize them? They are all having phosphoglyceride as their backbone, but one can be differ with other based on their head group. If the head contain choline, it will become phosphatidyl choline. If the head contain inositol, it will become phosphatidyl inositol. Now, that is the first type of the membrane lipids, phosphoglycerides. Compared to the phosphoglycerides, this is more important, everyone focus. The second one, we call it as sphingolipids. Okay, this is also a question which is asked in the Gurukul DL paper. Okay, listen, sphingolipids. When you observe the sphingolipids, sphingolipids are derived from a backbone called as sphingosine. You people no need to remember the structure, but it is very important to understand the structure. What is sphingosine? How can I define the sphingosine? Everyone observe. When you look into the sphingosine, it contains a long hydrocarbon chain, nothing else is present, hydrocarbon with a polar domain with an amino group. That's all. How can I define the word sphingosine? Sphingosine is a long hydrocarbon tail with a polar domain which is associated with an amino group. Sphingosine, which has a long hydrocarbon tail, a polar domain which includes an amino group. This is the simple structure of the sphingosine. Now, no need to get confused, understand one more time. You understand what is called sphingosine. Now, every student has to understand what is called ceramide. They are very, very important, focus correctly. What is sphingosine? Long hydrocarbon tail, polar domain with an amino group. If the amino group of the sphingosine form an amide bond with the carboxyl group of the fatty acid, that is what exactly we call it as the ceramide. Very simple. The amino group, this is the amino group. If it forms an amide bond with the carboxyl group of the fatty acid, that structure is called as ceramide. That is very important. I want you to remember especially ceramide and how does it formed. Now don't confuse. Everyone focus on this picture correctly. Now tell me what it is. This is ceramide. Correct na? Sphingosine. Till here it is sphingosine. Correct or not? Now sphingosine formed what bond? Amide bond with carboxyl group of the fatty acid. We call it as ceramide. Now every student observe the picture very correctly. Till here it is the completely ceramide. If this ceramide having the head which is phosphocholine, this is the structure of the phosphocholine. If the ceramide having phosphocholine as their head group, we call it as sphingomyelin. We all say our neurons contain what sheath? Myelin sheath. Correct na? Myelinated neurons can easily protected from chemical and mechanical injuries. So that sphingomyelin is generally made up of phosphocholine plus ceramide. In very simple words, if ceramide contain phosphocholine PC as their head, that is what we called as sphingomyelin. Now what is the question which is asked in the Gurukul DL exam? This one. Okay, there are two type of sugars, everyone remember. One is called as the cerebrosides and another one is called as the gangliosides. What is the difference between the cerebrosides and gangliosides? How can I give the simple definition? I can give a very simple definition to all of you. Listen, I am writing here. If a ceramide, everyone understand, ceramide combined with a simple sugar. What is simple sugar? Mostly monosaccharide. Let's consider glucose or the galactose. When the ceramide combined with the simple sugars, we can call it cerebrosides. When ceramide combined with a complex sugar such as oligosaccharides or polysaccharides, we call it as what? Gangliosides. Cerebrosides and gangliosides collectively called as glycosphingolipids. We also call it as glycolipids, which are predominant on the neuronal membrane, right? And if you look into the inner membrane of the chloroplast, even inner membrane of the chloroplast also contain glycosphingolipids only, along with the sulfolipids. We'll see that. Now I am summarizing all here. Whomever having the doubts, please look into this very correctly. We are talking about the sphingolipids. You no need to focus on the structure, don't worry. Instead of the structure, focus on the structural features. If you observe the sphingosine, what does it contain? It contains a long hydrocarbon tail, a polar domain with what group? Amino group. This is called sphingosine. What is called ceramide? When the sphingosine amino group formed an amide bond with the fatty acid carboxyl group, we call ceramide. And what is phosphocholine? What is sphingomyelin? Whenever this is ceramide having phosphocholine as their head group, we called as sphingomyelin. Now every student look into the picture. Whenever the ceramide contains simple sugars such as glucose, galactose, monosaccharides, we call it as cerebroside. Whenever the ceramide contain complex sugars, we call it as gangliosides. Now friends, every student remember, there is no need to confuse with this statement. Understand what exactly I am trying to say. Many bacteria, whenever they enter in our body, in order to show their action, they have to attach us to the receptors. Best example, you take a cholera toxin. Cholera toxin caused by Vibrio cholera. Correct na? Food poisoning caused by Clostridium botulism. Correct na? So all whichever causing the cholera, whichever causing the food poisoning, all these bacteria, in order to show their action, they will release what? Their toxins. They are generally neurotoxins which impact on the nervous system. Those toxins will use ganglioside as their receptors. They use as what? Ganglioside as their receptor. So even though they are lipid molecules, they are acting like what? Receptors for most of the toxins that are entering in our body. No need to get confused over there. Now we understand, listen everyone, phospholipids and sphingolipids and glycerophospholipids. After that, another very important one is cholesterol. Now every online people answer the question, do you really think cholesterol is present in the animal cell or not? Every student answer, cholesterol is present in the animal cell or not? Now everyone answer, cholesterol is present in the plant or not? Plant. And the cholesterol is present in the bacteria? Cholesterol is present in the fungus, yeast. Remember, compared to all the organism, cholesterol is abundantly found in the animal cell. But when we consider the plant cell do not have the cholesterol. Instead of the cholesterol, they are having variety of other lipids. You taken a best example of the bacteria. Bacteria, cholesterol is completely absent. Apart from the cholesterol, they are having the other type of the sterols. So compared to all the organism, especially the higher animals, cholesterol will be present. Undoubtedly, cholesterol is present doesn't mean that the cholesterol is present in the higher percentage. Cholesterol is present in the very lower percentages only. Now, I want all of you to understand the picture. Correct, everyone correct. Suppose if I ask you a question, how does the lipid look like? You will say that lipid containing hydrophilic head and lipid contain what tail? Hydrophobic tail. How does it look like? It is looking a linear form, correct na? Linear form means straight form. Cholesterol, even though it is a lipid, it never be linear form. Then cholesterol is what form? Cholesterol is a ringed structure. What structure? Ringed structure. Now the ring of this cholesterol said to be cyclo, you have to remember this, pentano, perhydrophenanthrene. This is the ringed structure of the cholesterol. Cyclo, there are 5 rings are there. Cyclo, pentano, perhydrophenanthrene ring. That is the ring structure of cholesterol. Can everyone focus on the picture? Can you see here? Cholesterol contain what group? OH group. We can call this is called head group. And what about this? This is called as tail group. Now all of you focus, how does the cholesterol embedded within the plasma membrane? Look into the picture correctly. Cholesterol head having what group? OH group. Now can you see here? The OH group of the cholesterol, in other words, the head group of the cholesterol can associated with the head of the lipid. Can you see that? Similarly, this part is what? Tail part. So tail part of the cholesterol can associate with the tail of the lipid. In very simple words, friends, head of the cholesterol can fuse with the head of the lipid, or hydrophilic part associated with hydrophilic part, hydrophobic part associated with the hydrophobic part. That is how cholesterol will be completely embedded within the plasma membrane. What are their functions? We will read in the second part. Cholesterol majorly responsible for what? The fluidity of the membrane. Means movement of the lipids. Lipids has to move continuously on the membrane in order to maintain their fluidity. So far whatever we have discussed till this particular slide, the first major composition of the plasma membrane, lipids. We may get the question, we may not get the questions, but we have to read the lipid composition compulsory. When compared to the lipid, protein composition and carbohydrate composition is more important towards the exam point of view, where we can expect the direct questions. Now, membrane proteins. Okay, good afternoon. Is it clear everyone so far? Please confirm me. Everyone is clear till this particular lipid part? All of you please confirm. All right. Now, as you all people given the answer, proteins are the one which determine most of the membrane's specific functions. Correct now? Now here comes the question, how many types of proteins are present? So on the entire plasma membrane, only one type of protein or two type of protein, how many types of proteins are present? Again, that is also completely different from the type of the cell. If you generally consider a plasma membrane is a collage of a different proteins, and interesting point is that most of the proteins whatever present on the membrane, they never arranged separately, they always arranged or grouped together. That's the reason we always call it as a collage or a group of proteins. Now, this is also a very important question. Everyone has to remember this correctly. Now, you all people given the answer right. Proteins are the one which are performing the major functions. What kind of functions that the proteins are performing on the plasma membrane? Number one, they are acting like what? Transporters. Tell me, on the plasma membrane what we are having? We are having channels. They are called channels. They are called as what? Pumps. Some are called as carriers. So what are the major functions of these channels, carriers or pumps? What are their major function? They are involved in the transport of ions or transport of molecules across the membrane. Now when you observe chemically, these channels and carriers which are acting as a transporters, they are basically proteinaceous in nature. Now, some of the proteins which are present in the plasma membrane, they will be acting like a linkers. What is that? Linkers. What is the major function of a linker? Which is connecting extracellular matrix to the cytoskeleton. This is a cytoskeletal protein. What kind of proteins are present in the cytoskeleton? Cytoskeleton. Cytoskeleton contain microtubules, actin filaments, is it? So, cytoskeleton, or in other words, intracellular environment connected to the extracellular environment via linker molecules. The linker molecules are also chemically what in nature? Protein in nature. No need to confuse. And you also know very well, we read multiple times in the human body, whether it is a hormone, whether it is a growth factor, whether it is a neurotransmitter, whatever it may be, any molecule wants to work, what do they require? Receptor. Correct na? What is receptor? How can we define? Receptor is a specialized protein present on the plasma membrane which provides the binding site at which the ligand or the signal molecule will come and attaches. So this receptor also chemically what? Protein in nature. And you know very well, we cannot imagine any reactions without enzymes. What are enzymes chemically? Enzymes chemically or proteins only, right? All the enzymes are proteinaceous in nature. So the proteins which are present on the membrane acting like a transporters, some are acting like a linkers, some are acting like a receptors, and some are acting like a enzymes. These are all the major functions performed by the proteins present on the membrane. Now here comes a very important question. You understand what type of functions performed by the membrane, protein. What kind of proteins are present on the membrane first of all? How many varieties of types of proteins are present on the membrane? Peripheral proteins, integral proteins, then the third one we call it as transmembrane proteins. What is the meaning of a transmembrane proteins? Which transpan or which moves on the plasma membrane. They are called as transmembrane proteins. Now here comes a question, very important. How we categorized them into peripheral, integral, transmembrane? How did we categorize? We categorized them into different types exclusively based on their arrangement. Number one. Then of course based on the function performed by them. Major criteria is their arrangement. What we discussed, where the peripheral proteins are present. Suppose this is the plasma membrane. Peripheral proteins are present only at the periphery region. So their arrangement is completely different. Where the integral membrane proteins are present, they are embedded within the membrane. Where the transmembrane proteins are present, they are moving on the membrane. So every protein arrangement is completely different. So that's the reason, based on their arrangement, we categorize it into peripheral, integral and transmembrane. Now again we can get the question directly in the exam. What question we can get directly in the exam? Among all these proteins, which proteins are difficult to isolate by using the mild acids? Now can any one of you answer the question? Among the peripheral, integral, transmembrane protein, which proteins are very difficult to isolate from the membrane, separate from the membrane? And so we will see that. Very good. It is a basically integral type. Okay. So why we are calling integral membrane is the very tough to separate. Why not the transmembrane? Let's understand. Which can easily separate it? Why we can easily separate? So if you observe the peripheral protein, what we discussed, peripheral proteins are located here. Correct na? They are attached to which part of the lipid? Can you see here? Hydrophilic head. Correct na? Now friends, what is this linkage? This is important here. It is attached to the lipid via covalent linkage. What is that? Covalent linkage. This linkage is the covalent linkage. We can break this covalent linkage. It is associated via non-covalent linkage, not the covalent linkage. What are non-covalent linkages? Weaker interaction. You can consider hydrogen bonds. You can consider electrostatic interactions. You can consider hydrophobic interactions. So hydrogen bonding, hydrophobic, electrostatic interactions are very easy to break. Hence, in order to separate the peripheral protein, we no need to use any strong acids. We no need to use any strong detergents. Even by using a mild treatment, mild acids, we can easily separate them. There is no need to get confused over there. So proteins that are attached to the either surface of the bilayer, we call peripheral protein. They are associated with the polar head group of the lipid via non-covalent interaction, such as electrostatic interactions and hydrogen bonding. There is no need to get confused. Now, very important, everyone has to remember why we cannot separate integral membrane protein. Can you observe this picture? This is very important. This is the plasma membrane. Can you see here? This is the integral membrane protein. Integral membrane protein embedded within the plasma membrane. Now, you know that the plasma membrane contain head, plasma membrane contain tail. Now, how this particular protein embedded, last time we discussed in the biochemistry, this integral protein contain hydrophilic amino acids also, and it also contain what? Hydrophobic amino acids also. You know, right, what is this structure? Secondary structure of the protein. What are these? Alpha helix. Correct na? We call alpha helix and beta pleated structures. Now what I am trying to explain to all of you, this is the integral protein made up of both hydrophobic and hydrophilic amino acid. Hydrophobic amino acids which are present in the this integral protein connected to the lipid bilayer. Hydrophilic amino acids present in the protein connected to the hydrophilic head. So hydrophilic and hydrophobic amino acid help the integral protein to associated with the membrane, and it is always deeply integrated within the membrane. So if I want to separate this, I should always use stronger detergents, organic solvents such as ether, chloroform, carbon tetrachloride and denaturants. When we use all these powerful substances, then only we can separate them very easily. Why? Because most of the integral proteins are associated with the plasma membrane via hydrophobic interactions. So in order to separate these hydrophobic interactions, mostly we have to use strong detergents. Detergents are most effective in removing the hydrophobic interactions. Now, everyone understand this statement. This is also a very important statement. I want you to understand how a protein is associated with the plasma membrane. How a protein is associated with the plasma membrane. This is very, very important. Can you see here, whatever the protein that is present on the plasma membrane, it is transpanning on the plasma membrane only one time. What does it mean? We all read in the set and net also, right? It is called single transmembrane protein, which means which is transpanning on the plasma membrane only one time. We read now there are some proteins transpan 7 times. What is the name of that protein? We read now unit 4, cell signaling. We call it GPCR protein or G protein. They will transpan the plasma membrane 7 times. What happened to some proteins? All the proteins come close together, they can form a channel like structure, or they can form a pore like structure. Can you see that? This is called as a pore like structure. Best example what you people can give, you can give aquaporins. Correct na? What are aquaporins? Water channels. Why we call pore? Because all the transmembrane proteins come close together, they can form a channel or a pore like structure. That's why we call it as the aquaporins. So whatever the proteins present on the membrane, sometimes they transpan the membrane one time, sometimes they transpan the membrane 3 times, sometimes they transpan the membrane 7 times, sometimes they transpan the membrane 10 times, depending on the protein. Sometimes all the proteins come close together and they are going to form a channel like structure. No need to get confused. Can you see this picture? There is no need to get confused. This is a integral protein or a transmembrane protein. This is a peripheral protein. Correct na? Sometimes what happen, one protein can be associated with another protein. This is called protein-protein interactions. This is how the proteins are associated with the plasma membrane. No need to get confused with this particular point. Now, this is the most important statement that all the students has to understand. Okay. All transmembrane proteins are integral membrane proteins, but not all integral membrane proteins are transmembrane proteins. Tell me everyone, how can you justify this particular statement? Tell me. They are using a word, all transmembrane proteins are integral. Let's consider this is the plasma membrane. This is the transmembrane protein which is transpanning the membrane. If it wants to move on the membrane, if it wants to transpan on the membrane, first it has to embed it within the membrane. So since it is embedded within the membrane, we call it as integral protein. Correct na? So that's why every transmembrane protein is integral membrane protein. Now let's consider this is the integral membrane protein. It may transpan the membrane, it may not transpan the membrane. Then what we can consider? Not all integral membrane proteins are transmembrane proteins. So that is a very important statement to be understood. Now this is also a very important question. I want all of you to remember for the exam. There is a great possibility we can expect the question. There are 6 types of integral proteins are present on the membrane. How we categorize these integral proteins into 6 types depending on their orientation, which means how they are arranged on the polypeptide. Based on that we categorize it. This is so important. I encourage everyone to remember that compulsory. Type 1, type 2, type 3, type 4, type 5 and type 6. There are total 6 type of the integral proteins are present. There is no need to get confused over there. Now, how one can be differ with other? This is very, very important. Everyone, I want everyone to focus on the picture so clearly. Let's consider type 1 integral protein. when we observe the type 1 integral membrane protein, can you see the picture, friends? It is transpanning the membrane only one time. Type 1 integral protein transpan the membrane one time. Can you see the interesting point here? When you observe type 2 integral membrane protein, type 2 integral membrane protein also can transpan the plasma membrane only one time. Now here every student has to answer me now. If if both membrane, I mean to say, if type 1 integral protein, type 2 integral protein, if both are transpanning the plasma membrane only one time, then what is the difference between the type 1 and type 2? That's a very good question. If you observe the type 1 integral membrane protein, every student focus, N-terminal domain is located outside, C-terminal domain located inside. Friends, but when you observe the type 2 integral membrane protein, C-terminal domain located outside, N-terminal domain located inside. This is the fundamental difference between the type 1 integral membrane protein and a type 2 integral membrane protein. Once again, every student remember, there are 6 type of integral proteins are present on the membrane depending on their arrangement. When you consider the type 1 and when we consider the type 2, both type 1 and type 2 transpan the membrane only one time. When you consider the type 1, N-terminal domain located outside, C-terminal domain located inside. When we consider the type 2, C-terminal domain located outside, N-terminal domain located inside. This is what, friends, type 2 integral membrane protein. There is no need to get confused over there, and it is easy to understand. Now friends, everyone focus on the picture. If you look into the type 3 integral membrane proteins, you have to remember the properties compulsory for the exam. If you look into the type 3 proteins have multiple transmembrane helices. What does it mean, friends? Because I want every student has to remember the difference between the type 3 and type 4 compulsory. Type 3, what I am saying, type 3 proteins have multiple transmembrane helices in a single polypeptide. What does it mean exactly? Every student please look into the picture correctly. If you observe the type 3, it is only one protein, but it is transpanning multiple times. An example, you can take a GPCR only. Type 3 integral membrane protein, only one protein transpanning multiple times. But don't confuse with the type 4. Everyone understand. When we consider the type 4 proteins, they are all not single polypeptide. This is different, this is different, this is different, this is different, this is different. What you have to understand, different polypeptides come close together and they are going to form a channel-like structure. This is a very important statement I want every one of you to remember for the exam. Type 4 proteins are the transmembrane domains of several polypeptides assembled together to form a channel through the membrane. This is what exactly we call it as the type 4 integral membrane protein. I'll quickly summarize. Type 3, it is one polypeptide transpan multiple times. Type 4, different polypeptides coming close together, they can form a pore or a channel-like structure. Very simple to understand. Now friends, when you look into the type 5, it's very simple. Can you see this picture? Type 5 is a type of a protein which is connected to the plasma membrane via lipid linkage. Very simple. Focus all of you. Type 5 proteins are held to the bilayer primarily by covalently linked lipids. There is no need to get confused over there, and it is easy to understand. And when you consider the type 6 proteins, this is very important. All the students focus on the type 6 protein very correctly. Type 6 protein, it is having the transmembrane domain also, very clear, and it is having lipid linked domain also. What you have to understand, Type 6 protein attached to the plasma membrane via two domains, transmembrane domain also as well as the lipid linked domain also. These are the total 6 type of integral proteins present on the plasma membrane. It's very important. I want you to remember this very clearly, friends. Without this you can't understand anything, right? This is not required. Now what we understand so far is type of proteins present on the plasma membrane, type of lipids present on the plasma membrane, and their major functions. Now apart from the lipids and protein, what else the plasma membrane contains? The plasma membrane majorly contain carbohydrates apart from the lipids and proteins. Now do not think that compared to the plasma membrane and compared to the lipids and proteins, carbohydrates never present in the highest percentage. Compared to the proteins and lipids, carbohydrates always present in the lowest percentage, very lowest. I can say hardly it is present only in the 5 to 7%, that's all. 5 to 7% of the plasma membrane contains carbohydrates. Now I am requesting all the online students, give me this simple answer. What is the major function of the carbohydrates on the plasma membrane? What are the major functions of the carbohydrates on the plasma membrane? Any one of you? I want you to tell me major function. Major function of the carbohydrates on the plasma membrane. Any one? Any one of you? Sudarshan Reddy, tell me what is the major function? Receptor, okay. Anyone else? What is the major function of the carbohydrate that are present on the plasma membrane? Everyone remember the very good, very good, Shiva. The major function of the carbohydrate is cell-cell communication or the cell-cell attachment, or you can also say cell signaling also. Carbohydrates plays a very crucial role in the cell-cell communication. Now I will explain very briefly about the carbohydrates. I want every one of you to be much patient and try to understand it very clearly. Okay. Now all of you pay attention and try to understand this. Compared to the proteins and lipids, carbohydrates are present only in the very low percentage. What I told to all of you, whatever the carbohydrates that are present on the plasma membrane, they are associated with lipids also, and carbohydrates are also associated with the proteins also. But what I told to all of you, compared to the lipids, most of the carbohydrates, they are associated with the proteins only. That's the reason what we can say that. We can say very clearly carbohydrates are most predominantly present in the form of a glycoproteins. That is a very important statement. I want you to remember here very correctly. Okay. Now what is this process called as? Every student listen. The attachment of the protein to the carbohydrate, this process we generally call it as glycosylation. Glycosylation is a process of addition of the sugar to the protein for the cell communication. We call it as glycosylation. It is extremely responsible for what purpose? It is extremely responsible for cell recognition. Now let's understand very elaboratively. This is also a question 90% you people can get in the exam. Very simple and very important question I want you to observe. This is a sugar, this is a protein. Everyone understand the point very correctly. How does sugars can be attached to the protein? What is the type of a linkage? All the sugars can be attached to the protein via only two type of a linkages. One is O-linked glycosylation, then another one is called as N-linked glycosylation. Why we are calling it as a O-linked glycosylation, which means sugars are attached to the protein via oxygen as their major linkage. And what is called N-linked glycosylation? Sugars are attached to the protein via nitrogen linkage. That's why we can call it N-linked glycosylation. What kind of questions you can expect in the exam? Variety of question. An example, which is the site or which is the location of the N-linked glycosylation? Every student remember the point. N-linked glycosylation exclusively takes place in the endoplasmic reticulum lumen. What is called lumen? Inner space. So at the inner space of the endoplasmic reticulum, we will get we will have N-linked glycosylation. Very important. Where does the O-linked glycosylation will be happening? O-linked glycosylation is exclusively happening in the Golgi complex. There is no need to get confused, friends. O-linked glycosylation happen in the Golgi complex. N-linked glycosylation is exclusively happening in the lumen of the ER. Now here comes a very good question. What kind of amino acids involved in the O-linked glycosylation? What kind of amino acids are involved in the N-linked glycosylation? This is important for the exam. All the students, please focus on the picture. Every one of you. This is a protein. This is a protein. Every student know that proteins are generally made up of what? Proteins are generally made up of amino acids. So in this particular protein, there are two amino acids, that is serine and threonine, are present in the active site. Every student know that serine and threonine, what groups they are having? They are having OH groups. Correct na? Serine and threonine having OH group, exactly to this oxygen the sugars will be adding. Can you see that here? That's why it is called O-linked glycosylation. So in the O-linked glycosylation, the type of amino acids involved are serine and threonine. Very easy to understand. Then when it comes to the N-linked glycosylation, so again this is the protein which is made up of amino acid. In the active site of the protein, the amino acid that is present is the asparagine amino acid. What is that? Asparagine amino acid. Asparagine amino acid having what? Amino group. So exactly to that nitrogen the sugars will be adding. So in a very simple words, friends, in the O-linked glycosylation, serine and threonine will be added. In the N-linked glycosylation, asparagine is the amino acid that is involved. Okay. Everyone, please confirm me, online people who are attended so far. Is it clear for everyone? Is it clear for everyone? Everyone, please confirm, all the online people who are attending. Clear for everyone so far? If anyone having any kind of a doubt, you can ask me. Okay, now all the students listen to me very carefully. From Monday onwards, everyone listen. From Monday onwards, the class timings, okay, Monday, Tuesday, Wednesday, everyone please listen. Monday, Tuesday, Wednesday, these 3 days all the classes will happen through the app itself. After that, whomever enrolled for the course, all the classes strictly happen through the app itself. Only up to Monday, Tuesday, Wednesday, all the classes will happen through the YouTube. Okay, as the demo classes, remaining all the classes will happen through the app only for the paid users. And remember, from the Monday, the class timings are extremely fixed. Okay, 5:15 AM, okay, to 6:30 AM, sorry, every single day. Okay, Monday to Saturday. Monday to Saturday, the timings are very fixed, 5:15 AM to 6:30 AM. Then when it comes to the evening time, okay, 4:30 p.m. to the 6 p.m. So every single day we will be having 2 point how much it is, this is 1.5 hour, right? Okay, so every day we will be having 2 hour 45 minutes class. Okay, Monday to Saturday. Okay, Monday morning for botany people and for zoology people, I will give a separate timetable day wise. Which day, which concept will be covered, and when you will be having the test, chapter wise test, and when you will be having the mock test, everything I will provide you in the Monday morning 5 a.m. class, 5:15 a.m. class itself. Okay, complete botany timetable till the end. We will consider, everyone remember, we will consider June 5th or June 6th, maximum we will consider June 6th is the last date. June 6th is the last date for syllabus completion. Okay, in this only I will include the test series also, but mock test series we will start from June 1st to 10th. Okay, so by June 6th I will cover complete syllabus. I am giving you the assurance, out of 100% we will cover the syllabus up to 90 to 95% of the syllabus, and that is completely sufficient to crack the exam. So if anyone is really interested, really passionate, then only you enroll. Please don't enroll for time pass, okay, because I have to put a complete effort for all of you. So if I am expecting the same kind of effort from other side, then only it will be good. If you consider Telangana result of the Gurukul DL, TSPSC, JL, every single student who are qualified exclusively from our institute only. So however the efforts they kept during their preparation, if you are ready to put that kind of preparation for your APPSC and upcoming TGPSC, then only you can crack it. Syllabus completion, test conduction is our responsibility. Okay, your responsibility is working extremely hard from the day one. Okay, so Monday, Tuesday, Wednesday classes will be in YouTube. From Thursday onwards, whomever paid, classes will happen strictly through the app. Okay, I hope it's clear for everyone. If anyone having any doubt, you can WhatsApp us or you can call us, and please try to enroll by Wednesday. Okay, after that Wednesday I don't take much admissions also. Even though it is a 30-40 students, there is no problem, we will continue the batch. Okay, but when it comes to the book, as I already told you, only 1000 copies are available. For enrolled students, there is no need to pay separately for the book. Whomever only wants the MCQ book also, you can order by April 10th. Okay. Thank you all. Thank you.